All right, BIM 100 students, let's get you ready for that final. This is your study guide for Lecture 1. We're going to go over the key concepts and, more importantly, some pro tips for how to approach Professor Pasquinelli's questions.
Okay, so here's our game plan. We'll kick things off with those classic experiments that proved what the genetic material is. Then we'll hit a super important exam tip about interpreting data. After that, a quick review of DNA structure, then another exam tip, this time on melting temperature problems, and we'll wrap it all up with the key takeaways you need to know.
First things first. Let's do a quick memory refresh on the Hershey-Chase experiment. This is the one that really sealed the deal and proved that DNA is, in fact, the genetic material. Now, you're pretty likely to see a question on the final that's something like this, asking you to explain the logic of this foundational experiment. So let's break down exactly how they did it.
Okay, let's walk through this famous blender experiment. It's actually pretty clever. They took 2 batches of bacteriophages. In one, they tagged the DNA with radioactive phosphorus, P32. Why phosphorus? Because DNA has it and proteins don't. In the other batch, they tagged the protein coats with radioactive sulfur, S35. Why sulfur? Because proteins have it and DNA doesn't. They let these phages infect some bacteria, threw them in a blender to knock the phages off the outside of the cells, and then spun them down. The big question was, what got inside the bacteria?
And boom, there's your conclusion. The only radioactivity they found inside the bacteria was the P32 from the DNA. The S35 from the protein coats, that was all left outside. So it's the DNA, not the protein, that gets into the cell and starts calling the shots to make new viruses. That's the punchline. Okay, let's switch gears to our first major exam tip. And honestly, for Professor Pasquinelli's class, this is a huge one. You have to know how to handle data that seems to go against everything you've learned. You probably remember this clicker question from lecture, right? It is the perfect setup for a classic Pasquinelli-style question. It tests whether you can interpret results directly, not just spit back facts you've memorized. So listen up, this is so important for the exam. The dogma says DNA is the genetic material. We know that. But look at the data here. When you treat the brain extract with enzymes that destroy RNA or DNA, it's still infectious. But when you use protease to destroy the protein, it's not infectious anymore. So based only on this experiment, your conclusion has to be that protein is the infectious agent. You answer based on the data in front of you, period. All right, let's do a super quick refresh on the basics of DNA structure. You gotta have this stuff down cold for the exam, so let's make sure we're all on the same page.
So the fundamental building block is the nucleotide. It's got 3 parts, a phosphate group, a 5-carbon sugar, and that nitrogenous base. You really need to be able to just look at a diagram and point out what's what. These are the little Lego bricks that build the whole thing.
And what about the difference between DNA and RNA? It's so simple, but you have to know it. It all comes down to one little oxygen atom on the 2' carbon of the sugar. In RNA, you've got a hydroxyl group, an OH. In DNA, it's deoxyribose. It's missing that oxygen. That's it. That's the whole difference right there.
And of course, the base pairing rules. A with T, G with C. But here's the part that really matters for the exam. The number of hydrogen bonds. AT pairs are held together by 2 hydrogen bonds. GC pairs are held together by 3. That extra bond for GC makes it a stronger connection. Remember that. It's about to become very important.
Which brings us perfectly to our next big exam tip, melting temperature, or TM. You are almost guaranteed to see a quantitative problem about this on the final, so let's make sure you know exactly how to crush it.
Professor Pasquinelli literally asked this question in class, which is a pretty big hint, right? The higher the GC content, the higher the melting temperature. Why? Let's break it down.
It all comes back to those hydrogen bonds we just talked about. It's simple, really. Since GC pairs have 3 bonds holding them together and AT pairs only have 2, it just takes more energy, more heat, to pull apart a GC pair. So the more GC pairs you have in a strand of DNA, the higher its melting temperature is going to be. Simple as that. So let's put this into practice with a question that's going to look super familiar on the exam. They'll give you the percentage of one base and ask you to figure out the percentage of another. Okay, don't overthink this. It's a simple 4-step process. First, remember Chargaff's rules. G always equals C. So if you have 30% guanine, you must have 30% cytosine. That's step 1. Step 2, add them together. 30 plus 30 is 60%. So 60% of your DNA is G and C. Step 3, the rest of it has to be A and T, right? So 100% minus that 60% leaves you with 40%. And for the final step, since A always equals T, you just divide that 40% in half. That means you have 20% adenine. Done.
All right, let's bring it all home. What are the absolute must-knows from Lecture 1 as you're hitting the books for the final?
To sum it up, when you're studying, don't just memorize facts. You need to focus on these skills. Can you look at a piece of data and draw a conclusion from it and it alone? Can you explain the logic of an experiment, not just its result? Do you know why GC content affects melting temp, those 3 versus 2 hydrogen bonds? And can you do that quick math with the percentages? If you can nail these skills, you'll be in great shape for this part of the exam. And I'll leave you with one last brain teaser to chew on, something to really test your understanding. How is it possible that a cow and a salmon, 2 totally different creatures, could have the exact same DNA melting temperature? Well, if their melting temps are the same, that means their GC content is the same. So what makes them so incredibly different? It's all about the sequence, the order of those letters. Okay, good luck with your studies. You've got this.